Okay, so your packet asks what is hemoglobin, and it gives you a bit of a clue and expects you to look it up in the internet or in the textbook. If you take a look at hemoglobin, it's found in red blood cells. It carries oxygen from your lungs to your tissues. It's by concentration, so where there's high concentration of oxygen, it binds to the hemoglobin, and where there's low concentration of oxygen, the hemoglobin releases the oxygen. And then it diffuses into the tissue, which we've already talked about diffusion and osmosis, so hopefully you understand that. The hemoglobin molecule is made of four chains of polypeptides, four polypeptide chains. And together they form one molecule of protein. So it's a complex. There's two alpha chains. Alpha, this is a symbol alpha, and this is symbol beta, and two beta chains. These chains are, the reason they label them alpha and beta is just as a designator, just to let you know that this chain is different from this chain. These two chains are very similar. And we're going to look at normal hemoglobin, and then we're going to take a look at sickle cell hemoglobin, and that's really the key in your packet when you're doing those questions, is that you're looking at what is the change in the DNA structure that leads to the change in the mRNA that leads to this. What change does that lead to in the protein that actually causes this disease? Well, this is hemoglobin, and by the way, it's called hemoglobin because there's a group inside each of the proteins here that's called a heme. It is not a protein; it is a group, though. It acts as a cofactor, organic cofactor, and it associates with iron. It's the iron that actually attracts the oxygen. And in a way, it rusts. It's a weak rusting. It's a reversible rusting that happens. It's not really. It's oxidation, but it's not even a full oxidation. It's just an association of oxygen so that it pulls the oxygen in, but it's held loosely enough that when the oxygen is in a low concentration, the oxygen leaves the hemoglobin molecule and enters the cell. So that's really key. Here are a bunch of red blood cells. These are normal-looking red blood cells here. They'll have the little white center, which is it's white because it's actually the thinnest part of the cell. As you'll see later, as you saw earlier, and you'll see again in a minute, there's a concave kind of a dip on both sides. So this is the thinnest layer between the cells. When you look at an example of someone who has sickle cell trait, you'll see sickle cells in the blood samples when you look under a microscope. So these are normal cells, and these are the sickled cells. So there's a change in the DNA that changes the mRNA that then causes this sickle cell, this sickled shape to the cell, and that has implications. There are consequences to that, and those consequences can be deadly. And so let's talk a little bit about that. I think it's worth our time. Before we do look at the consequences of that, I want you to see this is a red blood cell here, and it has a little dip here, a little divot. The same divot's on the other side. So you see it's going to be the thinnest part of the cell is going to be right here. Now this, of course, is a sickled cell. The cell is oddly shaped. There's a benefit to having a sickled cell. There's a disease, parasite known as malaria, that kills millions of people, has killed countless numbers of people throughout history. It actually restricts a lot, and not just people, but cattle and other mammals, and it's a horrible little parasite. It's found in Africa and in the Mediterranean, anywhere where there's swamps or wetlands, and there's that more humid, warm temperature. So this parasite malaria it doesn't do a good job inserting itself into these sickled cells. These sickled cells resist malaria. And so someone that has sickle cell trait actually has malaria resistance, and therefore has an advantage over someone who has just normal red blood cells. And so that's one of the reasons it's so prevalent in populations where you have malaria as an issue in the environment. There is this related disease called thalassemia, which also is basically a sickled cell that gives the people with it a trait for it, also resistance to this malaria. The big negative, though, is that these cells cannot carry as much oxygen as normally would be carried by someone who has normal hemoglobin. So someone with the trait may have less, stamina than someone with normal hemoglobin, two normal hemoglobin genes. Now, the person with the normal hemoglobin gene, however, would then be susceptible to malaria. So the trade-off is that you get 1/4 of the population, having the disease and having a higher chance, greater chance of dying. You have 1/4 of the population that has the normal genes and has a higher chance of dying in malaria-ridden countries. And then you have half the population that's resistant to malaria and has this trait that doesn't give them as much stamina, but allows them to survive. So that's kind of a trade-off that evolution has given people with this trait. So here's where the problems come in as you're moving through. The red blood cells normally move through. You'll notice it has this little divot again. That's those thin places. If you look at it, you'll see that the round shape, allows it to enter the capillaries very smoothly, very easily. Remember, the capillaries are about 1 cell thick, so having the rounded edges and allow these things to move downstream, very much like those lazy river rafts that have those rounded ends and just bump along the side and keep moving. When you look at a normal red blood cell, you'll see that there's a thinnest part here. That's that white section you saw underneath the microscope earlier on in this presentation, and you'll see that the normal hemoglobin actually just kind of fills in everywhere. There's a little globular protein that's throughout the red blood cell, capturing as much oxygen as each of these cells can carry. But when you have a sickled cell, what they do is that because of their shape, they get stuck inside. They kind of catch on the sides of the capillaries and cause blockages. And these blood clots can cause trouble. They cause a lot of pain. They can cause tissue death to the brain or heart or lungs. They cause all kinds of other issues that can lower the life expectancy of someone. But today's treatments really do improve the life of someone with sickle cell disease. It's still a disease that you have to live with, and it has to be treated. It's a chronic disease, but again, in days past, it was a disease that was much more deadly than it is today. Still, it's an issue that has to be dealt with if you have sickle cell disease. Sickle cell trait, you have virtually no serious negative outcomes, so you really don't have a lot less of these blood clots, but it's an issue, all right? Now here we have the abnormal hemoglobin formed strands, and you see this is what they should look like, little globular proteins. And if you have the disease, what happens is that the hemoglobin forms these strands that pull the cell into this odd shape, the sickle cell shape. And so really what you have to worry about is that there's some kind of change in the hemoglobin, and it turns out it's in one of those 4 protein polypeptide strands. There's some kind of change in the hemoglobin that causes that shape and change. Change in the shape. Sorry. It turns out it's a very minor change. You'll see it when you look at it. I don't want to reveal it, but as you have in your packet the DNA sequence for the section of the gene that codes for hemoglobin that has that mutation. And then you have to translate that DNA sequence into mRNA, and then you have to use the codon triplet code to translate that mRNA into the amino acid sequence. So I want you when you do your comparison, you do your chart, please think about how many changes did it take to make this disease happen in the DNA, how many changes did it take in the mRNA to make this happen, and then ask yourself how many amino acids, what number of amino acids had to be different between this hemoglobin and this hemoglobin to make this cell sickle and cause so much trouble, or save a lot of people, depending on whether you have the trait or you have the disease. So go ahead and think about that as you're doing the packet. We'll talk about it in class. Hopefully everyone's watching the video so we have another week to study for this exam. Good luck, and I'll see you on Monday. Bye.